Now we are going to study biosensors. The father of biosensors is Professor Leland Clark. What is biosensor? Biosensor is an analytical device which converts biosignals into electrical signals. Biosensors are an analytical device which converts biosignals into electrical signals. Different types of biosensors are used for the detection of toxins in food industry. We can also use them in the agriculture to detect trace elements. We can use them in alcohol detection, in the medical Parkinsonism detection. We can use for detection of flavors in the food, also calcium levels in the milk. Fermentation of cheese, we use biosensors, and that's all. So the different types of biosensors are used for the detection of toxins in the agriculture. In the agriculture, detection of trace elements. In the detection of alcohol. In medical industry, detection of Parkinsonism. In the food, for detection of flavors, also for the detection of calcium in the milk. So these are the different types of biosensors. Now biosensor again. We will revise. What is biosensor? It's an analytical device which converts the biological signals into an electrical signals. Father of biosensors is Professor Leland Clark. He first discovered or invented a device called Clark electrode, which divides oxygen content. The first father of biosensor is Leland Clark, and the word biosensor is derived from Kamman. The word biosensor is derived from Kamman. The characteristics of biosensors are precise, accurate, reproducible. Biosensors are precise, accurate, and reproducible. They are cheap, small, portable. Also free from they should be free from electrical noises. The reaction of biosensor should be independent from physical parameters. They should also have linearity, sensitivity, selectivity, and stability. Linearity is nothing but maximum sensor output should be given. Sensitivity, specific sensitivity should be present. Selectivity means it should not interfere with other materials, and stability means it should be it should give maximum output for specific period of time, and the lowest quantity of sample can also be detected. So the ideal characteristics of biosensors are: it should be cheap, small, portable. It should be precise, accurate. Reproducible, cheap, small, portable. It should be independent from physical parameters. It should be free from electrical noises. It should have linearity, selectivity, stability, and sensitivity. Linearity is nothing but maximum sensory output should be given. Sensitivity, selective sensitivity there. Selectivity is it should not interfere with other materials. Stability is it should be stable, and small quantity can also be used for the detection. Now components of biosensors. The components of biosensors include analyte, bioreceptor, transducer, electronic display. So there are 5 components of biosensor: analyte, bioreceptor, transducer, electronic display. Analyte is nothing but it is a mixture of sample. The component which is required to be detected is known as analyte. This is our detecting point. After analyte, bioreceptors are present. The different types of bioreceptors can be used as enzymes, hormones, enzyme, cell, aptamers, DNA, nanoparts. Enzyme, cell, aptamers, DNA, nanoparts. The biosensor components are enzyme, cell, DNA, nanoparts, aptamers. The aptamer is nothing but single-stranded DNA or RNA. So this bioreceptor detects the analyte. Then transducer. So they detect the analyte in the form of like light, pH, temperature, etc. Light, pH, heat, temperature. These are the detecting things. After detecting these biological signals or like analyte, transducer is present. This transducer the components or like the transducer act as photodiode, thermistor, pH electrode, quartz electrode. So the things in transducer are photodiode, thermistor, quartz electrode, pH electrode. Photodiode, thermistor, quartz electrode, pH electrode. And these converts one energy to another form. Transducer converts one energy to another form, and the step between from biosensor to transducer is nothing but biorecognition. So the detected signals is recognized. So this is biorecognition. Next, from the transducer to next is electronic. Electronic converts the analog signals. Electronic converts the analog signals to digital. So this is signalization. The transducer gives converted signals which electronic forms into display. So this step is known as signalization. So transducer produce transducing signaling agent. After electronics display is present. So the process signal is displayed. This is quantifier. Biorecognition, signalization, quantifier. These are the components: analyte, bioreceptor, transducer, electronic display. This step is biorecognition, and this is signalization because due to transducer signaling agent, and this is processing. After process signal, quantification occurs. The bioreceptors can be enzyme, cell, aptamers, and DNA. Next, the transducer may be photodiode, thermistor, quartz electrode, pH electrode. So now advantages of biosensors. Biosensor used for thermometer, BP, glucose, all we can check all the temperature, thermometer, BP, sugar. Also result is rapid. We can have continuous response. This is highly specific and calibration is not needed. So the advantages of bioreceptors are temperature, BP, glucose, all can be detected. The signal can be rapid. We can use continuously, and calibration is not needed. It is highly specific. Now the different types of biosensors are, there are total 14 types of biosensors. So there are 14 types of bioreceptors: enzyme, whole cell, immunosensors, DNA biosensor, magnetic, thermal, potentiometric, conductometric, luminescence, optical, and conductometric over, blood glucose, electrochemical, physioelectric. So the types of biosensors are enzyme, whole cell, DNA, blood glucose, conductometer, potentiometer, luminescence, thermal, optical, physiochemical, electrochemical, amberometer, optical, magnetic, immunosensors. These are the types of biosensors. And the advantages, applications of biosensors are: biosensors are used in food industry, fermentation, medical field, environmental field, biodefense. Applications of biosensors are: there are applications in food industry, medical field, fermentation process, environmental field, biodefense. So the food industry, biodefense, environmental field, fermentation field, and medical field. In food industry, we can identify and quality. We can identify the quality of food. We can modify food. In medical field, healthcare and diagnosis. And in environmental, pollution detection. In biodefense, used for biowars. So the applications of biosensors are: in food industry for identification and quantification of food, in medical field, healthcare and diagnostic purpose, in fermentation purpose, in biodefense like for biowars, and in environmental field for pollution.